Hello Mrs. Ko, I'm Irvin and this is our group's video on the mammalian cell. A cell carries out many important functions for survival and there are different organelles in order to help them to do so. But before we get into the details, it is first important to know, what exactly is a cell? So in order to answer that question, a cell is the smallest unit of life. The cells are the basic building blocks of all living things. They provide structure, take in nutrition from food, convert those nutrients into energy, and carry out specialized functions. They are also found in both unicellular and multicellular organisms. Moving on, we'll be looking at the properties of a cell. All cells consist of a cytoplasm enclosed in cell membrane. Cells are known for taking in raw materials and then processing these materials to make new molecules. These molecules can be either used by the cell itself or transported to other parts of the body. Mammalian cells are known to range between 10 to 100 micrometers in diameter. The cells also contain the body's hereditary material, hence making it able to replicate. The human body is made up of trillions of cells, and these cells can be seen with a light microscope, or micrographs appearing as color images, or even an electron microscope which magnify objects up to more than 200,000 times, whereas micrographs come out black and white but can be artificially colorized. We'll now be looking at the organelles of a cell. Firstly, the cellular membrane, also called a plasma membrane, surrounds the cytoplasm in all cells. This membrane functions to give the cell shape and to keep the internal components confined and separate from the extracellular fluid that surrounds all cells. Plasma membranes consist of lipids, a fatty-like or wax-like substance, and proteins. The most common lipid found in membranes, known as phospholipids, contain a phosphate group connected with a glycerol group and two fatty acid chains. Phospholipids heads are hydrophilic, attracted to water, while the fatty acid tails are hydrophobic, not mixing with the water, making the molecule amphiphilic. They are all partially permeable, hence able to control the movement of substances in and out of the cell. The remaining part of the cell bound by the plasma membrane and surrounding the nucleus, also known as the protoplasm between cell surface membrane and the nucleus, is the cytoplasm. The cytoplasm consists of a jelly-like matrix known as the cytosol that holds up the other organelles of the cell, including the mitochondria and endoplasmic reticulum. Proteins, amino acids, and sugars used for growth and cellular reproduction are also contained within the cytoplasm. It is the site where several important cellular functions, for example, dissolving of cellular waste products and facilitation of movement. The network of filaments in the cytoplasm provides cell with shape. It is also able to conduct electricity. Next up, we have the mitochondria, also known as the powerhouse of the cell. The mitochondria are complex organelles that are enclosed by two membranes, the outer and inner membrane. The outer membrane has porins to allow smaller proteins in and protein complexes to allow bigger proteins in. The inner membrane is, however, highly folded and compartmentalized. Numerous invaginations of the membrane are called cristae, separated by crista junctions from the inner boundary membrane juxtaposed to the outer membrane. Cristae significantly increases the total membrane surface area compared to a smooth inner membrane, and thereby the available working space. The inner membrane creates two compartments, the region between the inner and outer membrane. Called the intermembrane space, it's largely continuous with the cytosol, and the more sequestered spaces inside the inner membrane called the matrix. The matrix is the site for ATP production, and ATP is produced during aerobic respiration, where the food substances are oxidized to release energy. The energy is used by the cell to perform cell activities such as growth and reproduction. The mitochondria is also only visible using an electron microscope. Now we have the nucleus, also known as the brain of the cell. This is because it regulates many of the activities in the cell, including transcription and DNA replication. The nucleus has a few structures, including the nucleolus, nucleoplasm, and nuclear envelope. We will first look into nucleolus. The nucleolus is a round proteinaceous structure located inside the nucleus. Its main purpose is to synthesize rRNA and assemble ribosomes. It is made up of three main components: the fibrillar center Fc, when transcription of rDNA occurs, the dense fibrillar component DFC, which contains fibrillin, and the granular component, which contains nucleophosmin required for ribosome biogenesis. Next up, we have the nucleoplasm. Essentially, it acts like the cytoplasm of a cell. It is completely enclosed by the nuclear envelope and it contains the chromosomes and nucleolus. This is also the site where nucleotides and enzymes are dissolved. Lastly, the nuclear envelope. The nuclear envelope is a double lipid bilayer membrane which includes the outer membrane and inner membrane. These two membranes are connected by nuclear pores. The outer membrane is linked to the endoplasmic reticulum, while the inner membrane has a nuclear lamina that binds to chromatin and other organelles of the nucleus. It also encloses the nucleoplasm. The space between the two membranes is known as the perinuclear space. The endoplasmic reticulum, ER for short, is a type of organelle that forms an interconnected network of flattened, membrane-enclosed sacs or tube-like structures known as cisternae. The membranes of the ER are continuous with the outer nuclear membrane. It is responsible for both synthesizing the proteins and the packaging of proteins in vesicles for transportation to the Golgi apparatus. There are two kinds of endoplasmic reticulum, which are the rough ER and the smooth ER. The smooth ER does not have any ribosomes attached to its membrane, making it more tubular than the rough ER. It is responsible for synthesizing substances such as fats and steroids and detoxification, which is the process where harmful substances are converted into harmless material. The rough ER, on the other hand, consists of a network of flattened spaces lined with a membrane. It is made up of cisternae, tubules, and vesicles. Its surface appears rough when viewed under an electron microscope, as there are small particles called ribosomes attached to the outer surface. Its outer surface is continuous with the nuclear envelope. They also transport made proteins to the Golgi apparatus. Next, the Golgi apparatus, also called the Golgi complex or Golgi body, is a membrane-bound organelle of eukaryotic cells that is made up of a series of flattened, stacked pouches called cisternae. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to targeted destinations. It is located in the cytoplasm next to the endoplasmic reticulum and near the cell nucleus and receives lipid and proteins as previously stated. While many types of cell contains only one or several Golgi apparatus, plant cells can contain hundreds. The cytoskeleton is a complex dynamic network of interlinking proteins, filaments present in the cytoplasm of the mammalian cell. Its primary function is to give the cell its shape and mechanical resistance to deformation. Through association with extracellular connective tissues and other cells, it stabilizes entire tissues. The cytoskeleton can also contract, thereby deforming the cell and the cell's environment, allowing cells to migrate. Moreover, it is also involved in many cell signaling pathways and in the uptake of extracellular materials, endocytes, the segregation of chromosomes during cellular division, the cytokine stage of cell division as scaffolding to organize the contents of the cell in space and in intracellular transport, and can be a template for the construction of a cell wall. Furthermore, it can form specialized structures such as flagella, cilia, lamellipodia, and protosomes. The structure, function, and dynamic behavior of the cytoskeleton can be very different depending on the organism and the cell type. Even within one cell, the cytoskeleton can change throughout association with other proteins, and the previous history of the network also provide a track-like system that directs the movement of organelles and other substances within cell. A centriole is a cylindrical organelle composed mainly of a protein called tubulin. A bound pair of centrioles surrounded by a shapeless mass of dense material called the pericentriole material makes up a structure called a centrosome. They are typically made of 9 sets of short microtubule triplets arranged in a cylinder. Centrioles are involved in the organization of the meiotic spindle and in the completion of cytokinesis. Centrioles are a very important part of the centrosomes, which are involved in the organizing microtubules in the cytoplasm. The position of the centriole determines the position of the nucleus and plays a crucial role in the spatial arrangement of the cell. They are, however, not present in all eukaryotes. They are also responsible for producing cilia during interphase, plus the ester and the spindle during cell division. Next up, the ribosomes, which are small round structures that are either attached to the membrane of the ER or lying freely in the cell. They are needed to synthesize proteins in the cell. The ribosomes attached to the ER make proteins that are usually transported out of the cell, while the ribosomes lying freely in the cytoplasm make proteins that are used within the cell. They are hence the key component in translation as they convert genetic code into amino sequences. Last but definitely not least, we have the lysosomes. Lysosomes are membrane-enclosed organelles that contain an array of enzymes capable of breaking down all types of biological polymers, including proteins, nucleic acids, carbohydrates, and lipids. Lysosomes function as the digestive system of the cell, serving both to degrade material taken out from the outside of the cell and to digest obsolete components of the cell itself. In their simplest form, lysosomes are visualized as dense spherical vacuoles, but they can display as considerable variation in size and shape as a result of differences in the material that have been taken up for digestion. Lysosomes thus represent morphologically diverse organelles defined by the common function of degrading intracellular material. Here is a list of references that we've used to collate all the necessary information in order to make this video. This video was brought to you by Irvin Lam, Jane Goh, and Yishan. Hope you enjoyed the video. Mrs. Koh, thank you very much.